So what this project is about really is my quest to understand biological systems. Who knows what this is? Anybody? Okay, this is the metabolic map of human metabolism. So this is everything that we'd want to know and characterize, all the enzymes, all the signaling molecules, all the substrates that we could ever care about. And if we fully understood this process across all cell types and in the whole organism, then we'd have a really great handle on how the human body is working and how biological systems are going. But for me, this is too complicated. I'm not going to do all the biochemistry here and measure all these molecules. That's too much work. So I'm just going to get rid of that and try to simplify it for everyone. And the lens that I'm going to use to kind of make sense of this is network biology. And at the core of network biology is a really simple pair of ideas, and that is the node and the edge. Nodes are these kind of discrete entities that we care about, and edges are the relationships between them. So to give you an example of how you might use this lens, here's some signaling for MAP kinase, and you can see that this little schematic has a lot of stuff going on. It has the phospholipid bilayer, it has some kinases, it has some phosphates, it has some gene expression and regulation, and some cellular outcomes. So this is actually, even while very small in scope, conveys a lot of information and is quite complicated. So to simplify it, to learn about this system and make it more tractable, we're going to break it down into nodes and edges, where the nodes are the signaling proteins in this pathway, and the edges are the regulatory steps in between them. And you can see that this has now been really simplified into a way that we can do some better form statistics on. So with this in mind, what's really left for us to do is find out how we can learn these edges. If the nodes are the genes that I'm going to study in my biological system, we have a pretty good idea of what all the genes are. Not complete, but still pretty good. So now the question is, how do we annotate functional pairs of genes to build up these systems? And in my mind, there are two main approaches to doing this at the gene level. And the first are by characterizing genetic interactions, which some of you in the audience have a lot of experience with. And this is a relatively simple idea where the combination of two or more genetic perturbations, A and B, results in a different outcome, a different fitness or phenotype than what would be expected based off of the additive combination of those perturbations. And this can be explored using combinatorial screening, and a genetic interaction score can be calculated by the difference between the phenotype for the pair of perturbations minus the two separately. And this has been really successful, most recently shown in this publication for all pairs of deletions in the yeast cell. And this has really given a phenomenal insight into cellular components and biological pathways in yeast. And since then, a lot of groups have been developing similar approaches for human cells, and there have been a lot of papers doing this kind of work. And this is clearly really powerful and is going to continue to be a really essential tool in understanding biological systems. However, there is a weakness that we have to overcome when we want to screen all pairs of human genes. And that weakness comes to, you can encapsulate with a simple calculation. If you just have one gene or one guide RNA that you're targeting, you might need 1,000 cells to perform that screen or that assay. If you really want to carefully study a gene, you might use 4 guide RNAs to knock down that gene and assess the phenotype. If you scale that up to all expressed genes, suddenly you're needing a lot more cells. Note the log scale on X. If you want to do all expressed pairs of genes in a cell, you'll need 400 billion cells. That's a lot of cells. Just to give some context for what that would be like, you'd need 140 3-liter flasks at a cost of $30,000 without any expense or consideration of the serum or other reagents that you would actually be the cost limiting factor in this type of experiment. So we're not ready for this yet. what else can we do? This leads us to the second way of characterizing pairs of genes, and that is known as co-fitness. Co-fitness is another relatively simple idea, which states that if two genes are essential or have a phenotype in the same cell types or under the same conditions across many of these cell lines or conditions, then they're likely involved in the same biological process. And this is achieved not through combinatorial screening, but parallel screening, many one-dimensional screens across different conditions. And this is assessed, very difficult to see under these circumstances, but this is assessed by taking a Pearson correlation of vectors A and B, which gives you a sense of how related these two genes are. So I'm going to go into this in more detail with an illustration of how this works. Here's this very simple gene pathway where the green, gold, and silver genes are all participating in the same pathway. Imagine that this pathway has variable importance for growth across different cell lines. By a CRISPR screen, or even in plates, you could assess the impact of knocking out all of these genes in the same pathway. What you'll observe is that if this pathway is not being used for growth in this cell line, when you knock out any of these components, you won't see much of a phenotype difference. If you knock them out in a cell type where this pathway is very important, then you'll have a severe fitness deficit. It doesn't matter which one you knock out, the same deficit will occur. Now, imagine you plot the phenotypes of the green gene versus the gold gene. Now you can see that the phenotypes are correlated, the severe fitness deficits are occurring in the same place, and the lack of phenotypic differences occurring in the same states, and this makes all of the points fall along the same axis or direction. Note that I described a continuous difference across cell lines, but the same outcome happens even if there are discrete differences across the cell lines. Imagine you have mutations knocking out either the green gene or the gold gene. Now that cell line can't use that pathway for growth. If you knock out additional genes in the same pathway, you won't observe any phenotypic difference. If you knock out genes in cell lines for which the pathway is still active and being used for growth, you will see large fitness deficits. And if you plot the green gene versus the gold gene again, even though there's no continuous differences, you'll get a correlation across different cell lines. Okay, so that's an introduction to co-fitness. This area has been heating up, getting a lot of attention. Hunter Fraser actually studied this relationship in yeast and learned about protein complexes in the yeast cell. But since then, a lot of groups have been extending this kind of work to human cells. And a big part of this is actually the release of hundreds of whole genome CRISPR knockout screens by the Broad Institute, including some people in the audience who are involved in that. And what you can see here is that they've plotted the gene essentiality profiles, the knockout effect sizes across all these hundreds of cell lines for all of these genes. And if you cluster these gene essentiality profiles, each row, you'll see that similar genes, or genes in the same protein complexes, have similar gene essentiality profiles. They're very correlated in function. So actually, you can learn a lot about a gene and its function just from its gene essentiality profile and how it's correlated with other genes. And in fact, you can get some pretty clean, distinct groups that correlate with separate biological functions. So when I saw this data set, I was really excited. I immediately went into exploring it myself and asked what we could learn from this really great data set. Here's the first attempt in my analysis at looking at this co-fitness phenotype. And the key to interpreting these plots is that one filled-in square equals one correlated pair of genes, and that is what I will refer to as one co-functional interaction. So each one of these filled-in things are a pair of genes that are significantly correlated with each other. So for all these three classes of genes, olfactory receptors, peroxisome genes, and spliceosome genes, you can see that there's definitely quite a few co-functional interactions. But if you're like me, if you see all these co-functional interactions across human cancer cell lines, you'll be confused. Why do these olfactory receptors show shared function? olfactory receptors are thought to be neutral and not play a role in cancer biology for the most part. So why is this all so filled in? It would be great if we could use this data set to focus on the cancer biology and maybe remove some of these other relationships that seem to be popping up. So the first goal of my work in this project is to improve how we detect correlated gene profiles using these large data sets. Here's the approach that I developed to try to correct and maybe improve the signal in these data sets. This data set starts out as a big giant matrix of genes by cell lines, where each entry represents the CRISPR knockout effect size. And what I'm going to do is I'm going to select some control genes, which are the olfactory receptors. Alternatively, you can choose unexpressed genes and get more or less the same result. And once you've chosen this subset of the matrix, you can perform principal components analysis on this matrix, get all the principal components, and by permutation testing, you can get the significant principal components that are going above the background rate. And I'm going to call these significant principal components the potential signatures of confounding, because they're showing significant signal even though these are null genes that we're not interested in. So what's going on with these signatures of confounding? Well, as it turns out, the majority of the variance in this matrix is explained by the first principal component. So for all olfactory receptors or unexpressed genes, you'll see that they're all co-varying along the same axis. They seem to have correlated effect sizes more or less in one single direction, although there are 4 other principal components that were significant. So when I saw this, I was very hopeful. Maybe the confounding is very simple and we can remove it, and once we take the correlation of the corrected gene profiles, we'll see an improvement. So let's see what happened. Remember, this is what the data set looked like before. Now I'm going to perform the correction and detect corrected correlations between genes. Here's the result of that analysis. You can see that, as expected, going from before and after, the co-functional interactions amongst the olfactory receptors are largely wiped out. There's only a few left, and those tend to be at places where olfactory receptors are very physically close in the genome. Fortunately, the signal at these peroxisome genes, which we think are functionally related, persists and is not wiped out by this correction. But most surprisingly to me, if you look at the spliceosome genes, suddenly we go from a very sparse architecture to many, many co-functional interactions being called. And this isn't a subtle fluke thing or a threshold thing. Many of these interactions go from being completely uncorrelated, totally flat, to showing very significant, large, unambiguous correlations afterwards.
So what was I don't understand what you corrected.
What did I correct for? I just took the covariance of the null genes of stuff that I'm not interested in, and I regressed those out of those signatures that explain the most variance in the control genes.
So what was the biological rationale for that?
Those control genes do not have the biologically interesting information that we want, so any variance in those genes is representing noise or artifactual signal. Yeah, so so maybe this looks interesting, but you don't believe me that this is a good idea. Maybe. What else can we do to confirm that this is actually a reasonable idea? Well, I showed you these gene essentiality profiles, and those seem to make sense. But another thing you can do is, instead of looking at the rows, you can look at the columns. And if you look at this matrix, there are perhaps 4 main types of cells or cell lines that might reflect, actual known different cell types. So one question I had was, if you were to cluster these columns after performing the correction, would you be able to predict the cell type of that profile better? Would your accuracy increase? And in fact, that is what I saw. The area under the receiving operator characteristic curve increased from 0.735 to 0.880 after performing the correction. And this suggests that we're getting this biologically interesting cell type specific signal after performing this correction. So at this point, I felt confident that this was going in the right direction. But what are these signatures of confounding? Where are they coming from? I can now say that I have a hypothesis, I've tested many different possibilities, but actually the explanation was sort of simpler than I originally thought. And it seems to be that determining the appropriate units or scaling for each CRISPR screen is non-trivial. Remember that all of these screens are conducted separately. They have different signal to noise in each screen, and the cells are different cell types and are handled differently. So when you get out the counts and you need to scale the effect size, it's really unclear how to do that in an unbiased way. And what I saw with the weights on my signature of confounding on the first principal component, which was the most important, the variance in the null genes highly correlated with the loading, which suggests that what's happening is that the units of the CRISPR screen are being rescaled according to the variance of the null genes, so that everything is effectively being renormalized. So that seems to be the biggest source of variation. All right, so this is my new idea, but what can we actually learn from co-fitness now that we have a much larger data set than ever before? I'm going to tell you about 3 applications. First, I'll talk about relating genes to cancer drug targets, because this is a cancer data set. TP53 is the most mutated gene in human cancers. There are many genes in this pathway and multiple drugs targeting genes in this pathway, such as nutlin-3, which targets MDM2, which directly regulates P53. Here is the data from my corrections shown to you as a heat map. Each row is a co-functional interactor of TP53, a separate gene, and each column is one of the hundreds of cell lines that have been profiled. The heat in the matrix is the scaled effect size, the scaled knockout effect size. And you can see each cell line is annotated with whether it's mutated for TP53 and whether it's sensitive to this nutlin-3 drug, which acts in the TP53 pathway. So if you focus on the heat map, you'll see that most of the large effect sizes for these co-functional interactors lies in these TP53 wild-type cells that are sensitive to the drug. This makes sense. If you've already lost TP53, if you already have a mutation, you aren't sensitive to subsequent knockouts in this pathway. You can also zoom in on specific co-functional interactors. Remember, MDM2 is a drug target. It's a deubiquitinating enzyme. It's one of its closest interactors is USB7. It's also a deubiquitinating enzyme. If you Google this protein, you'll see that actually it itself is a drug target, and multiple groups have been developing new chemotherapeutic agents targeting USP7. So this gives me confidence that this data set really gives you meaningful information about cancer biology, and that you could discover drug targets from this data set. Here's a more ambitious example of taking some known cancer pathways with multiple diverse drugs. Here's the TP53 PI3K pathway. Here's MAP kinase signaling, and here's some ERBB surface receptor signaling pathways. I asked a question of if I took the gene profiles that are most correlated with drug sensitivity profiles, would I be able to reconstruct this drug gene network? In fact, that worked surprisingly well. The genes that are known drug targets to these drugs tend to decluster and correlate with those drug profiles, and they formed 3 discrete units or modules of genes that are known to be involved in these signaling processes. So again, this data set seems to pick out these drug-target relationships. Next, I asked whether we could connect genes to curated biological pathways more broadly, not just drug targets. Here are some curated pathways of biological process, molecular function, or cellular component from gene ontology. If you look within these gene sets, they are enriched for co-functional interactions. You can also look at string, which is a large database of protein-protein interactions. This is similarly enriched around 4-fold. If you specifically filter to the high confidence string protein-protein interactions, you get a pretty massive enrichment over around 16-fold. And if you focus on this quorum manually curated gold standard for protein complexes, you get around a 30-fold enrichment, which is about as high as you could hope for. So high protein-protein interactions are massively enriched for co-functional interactions. So knowing that, I would like to understand how gene function relates to these protein-protein interaction networks. This is the mediator complex for transcription, and here are all the co-functional interactions amongst genes in this complex. And you can see it's very rich, very well connected, and really a success of this method. And what's particularly interesting about this case is that the specific domains of the protein cluster more tightly together than across domains, suggesting that different domains have different functions in different cancers. So when I saw this, I immediately asked if the distance, the path lengths amongst these proteins is also meaningful. Here's a little demo. Imagine that these are genes, the nodes are genes, and the edges are protein-protein interactions. One basic question is whether the rate of co-functionality for genes close in the protein-protein interaction is different from the rate of genes that are far away in this PPI network. In fact, if you look at genes that are physically interacting, they have a very high rate of co-functionality, 40% amongst really high-quality interactions. If you are separated by one intermediary, you still are quite enriched, and you decay in this very smooth fashion. Even if you have 8 edges separating different genes, that's still enriched above background. So there seems to be very broad, diffuse information conveyed through the protein-protein interaction network. So again, just to reiterate, lots of different processes are reported by co-fitness, and this is a very useful data set for understanding biological systems. However, I did refer to the high number of cells and high dimensionality of learning about biological systems, so perhaps we can use this data set to reduce the dimensionality of CRISPR screening. Here's my attempt at using a community detection algorithm to partition the genome into gene modules. You can see that there are 2 massive modules of core growth processes where knocking out any of these genes tends to result in a very stereotyped gene profile and are hard to tease apart. But I was encouraged that many key cancer genes are spread across many modules that seem to have distinct signatures. And if you look in these modules, they're actually enriched for cosmic cancer gene census genes, suggesting that genes that are important for cancer are spread broadly across this genome-wide network, and furthermore, a lot of them are clustering in sort of local hotspots. But perhaps the most important thing to be aware of in this clustering of genes is that we start from over 15,000 genes, and we end up with fewer than 500 gene modules. And this is really critical because once we're below 500, that's the number at which performing tests of all pairs of modules becomes practical in a CRISPR screening paradigm. Testing all pairs of 15,000, way too much. Testing all pairs of 500, maybe we can do that. What do these modules look like? Here's the NF1 gene community. This is the Mendelian disease gene associated with neurofibromatosis type 1. Here's NF1, and here are all the other genes in this little module. Spread 1 and spread 2 are known RAS signaling genes. So this pathway seems to broadly make sense given the biology of this gene. Let's zoom into this in more detail. I was curious about why this was forming a gene community, and it turned out that all of these genes have a very specific pattern in which they have large effect sizes. They occur in gliomas versus other cell types, and not only glioma cell lines, but NF1 wild-type glioma cell lines. If NF1 is already knocked out, and NF1 is one of the most frequently somatically mutated genes in the human genome, then knocking out any of these genes in that pathway has no effect. It's only the wild-type gliomas that are susceptible to gene knockout of any of these genes. So instead of testing all of these genes separately, maybe you can create a community score by taking the first principal component and aggregating all of this information. And we found that this is just as powered as taking the NF1 gene and would simplify the number of hypotheses you'd have to test. Let's look at the association of these community scores. We can see that cell lines that are annotated as CNS or skin derived lines have much larger community scores than other cell lines. And this makes sense because NF1 is associated with neuroectodermal lesions. So these are exactly the cell types that you would expect. And this is far from the only cell type community score interaction we found. There were dozens and dozens of these. So we can see that certain gene modules are specifically associating with specific cell types. All right, so to summarize, I think I've shown you that I have a new method for improving co-fitness data quality using PCA. I've shown some applications of co-fitness data. It's broadly enriched across many gene ontology categories, best for protein-protein interactions, and I've given you some ideas for how to decompose large, complex networks into gene modules. I think this work is really critical because the number of published CRISPR screens is going up and is only going to further skyrocket. We need to have methods that aggregate and correct for all the biases in these diverse screens and really synthesize all the knowledge, all the immense knowledge that's being created, going from just a handful in 2013 to over 500 mentions in 2018. So I would like to acknowledge my committee members, Hunter Frazier and Mike Bassick, my advisors, Jonathan Pritchard and Will Greenleaf, who's here today. And of course, people in the Bassick and Pritchard labs have been very helpful. I've bothered Michael Haney and David Morgans a lot over the years, so I'm very grateful to them. But I've also created an app. You can go to this URL and explore the data yourself, fully interactive, and you can annotate it with your own gene scores. And like I said, it is now published in Molecular Systems Biology. We made the cover art, so you're welcome to read it yourself. Thank you.